Good afternoon, one and all. And I'm Arun Balaji from the Faculty of Engineering, Architecture and Information Technology. Today I'm here to pitch you my research project idea entitled Self-Assembled Polymeric Micelle for Hepatic Fibrosis, an approach to non-invasive staging and targeted drug delivery. So, hepatic fibrosis is the common pathological feature of different liver diseases. Liver is one of the largest internal organ present in the human body. It performs hundreds of functions and plays an important role in maintaining homeostasis. Liver is made up of different types of cells, among which the hepatocytes alone make up nearly 70% of tissue mass. So, they are considered as the building blocks of the liver. But these hepatocytes are highly prone to damage, and it is mostly affected due to chronic consumption of alcohol and viral infections such as hepatitis. But these hepatocytes has excellent ability to regenerate, and this special cell population called hepatic stellate cells controls and initiate that regeneration mechanism. But in chronic conditions, instead of activating the regeneration of hepatocytes, the hepatic stellate cells get activated and transdifferentiate into myofibroblast like cells. These activated hepatic stellate cells start synthesizing excessive collagen fiber and transforms liver into a solid fibrous mass. This condition is called hepatic fibrosis. If unchecked, it leads to an advanced stage called cirrhosis. The cirrhosis leads to several other complications, including hepatocellular carcinoma, and then it ultimately leads to liver failure. Each year, the number of deaths due to hepatic fibrosis is increasing, and it also takes huge amount of money from health funds, which indicates hepatic fibrosis is one of the serious social and health issue. Despite better knowledge of pathophysiology and years of research, the current approaches are not effective in containing hepatic fibrosis. So, the basic research question this project asks is that, can we address the clinical complications associated with diagnosis and treatment of hepatic fibrosis using nanoparticles with targeting ability and non-invasive imaging techniques? Before I introduce you the core aspects of my research, let's see a few information about the shortcomings of current approaches. Liver biopsy is the gold standard technique used to diagnose and stage this hepatic fibrosis, but it has several disadvantages, and it's not applicable for continuous monitoring over a period of time because the patient may need to undergo multiple invasive procedures. The alternatives such as fibroscan and MRI gives you good results, but it's applicable to only few types of liver diseases, and it can't be used for doing the staging. On the other hand, the therapy applied for hepatic fibrosis also face several issues such as uptake by off targets and then low drug concentration at target and the inability of the drugs to pass through the distorted liver architecture. Because of these issues, we don't have any FDA approved drugs or treatment for hepatic fibrosis. So, how I am going to address these issues? Well, we know that in fibrotic liver, we have excessive amount of collagen fibers and activated hepatic stellate cell population. So, we hypothesize that by using nanoparticles which specifically accumulate in this target, we can rectify the problems associated with current approaches. Of these different surface receptor, we are going to concentrate on the receptors which overexpressed on the activated hepatic stellate cells, namely the mannose-6 phosphate receptor, and then the retinol binding receptor, and then the integrin receptor. So, these receptors will be targeted using nanoparticles which are conjugated to corresponding ligands. Further, a synthetic peptide which targets type 1 collagen and naturally available protein fragment which target type 1 and type 4 collagen will be used along with single fragment antibody to target the collagen content and also monitor the same. The nanoparticles I am going to use has imaging agents on its surface which enable us to monitor the level of the targets which I talked about using this high resolution hybrid imaging facility called PET MRI. So, for therapy, the hepatic stellate cells, the activated one, will be targeted specifically using the nanoparticles loaded with drugs which has been already conjugated with corresponding targeting ligands. And in order to increase the specificity, we will be using dual ligand approach, that is simultaneous usage of two different ligands on a nanoparticle system. So, how I am going to validate my research and what are the data I am going to obtain to do that? Initially, the physical chemical properties of the nanoparticles such as shape, size and surface will be studied extensively because they plays an important role in deciding the in vivo fate and the ability of nanoparticles to avoid the adsorption of plasma proteins. Later, the best combination of ligands will be identified through various in vitro and in vivo techniques for targeting hepatic stellate cells and the collagen fibers. And the amount of our targets present in the fibrotic liver will be monitored by using the contrast to noise ratio between the liver and the surrounding tissue. Then this information will be correlated with the corresponding histological data in order to demonstrate the accuracy of the PET MRI and the proposed imaging analytical method. So, PET MRI will be extensively used to perform non-invasive staging, and it will also play a central role with other imaging modalities to study the targeting ability, biodistribution and clearance of the nanoparticles. The in vitro studies of the nanoparticles will be run on different cell lines, and the rat models induced with hepatotoxin induced fibrosis will be used to study the will be used for the in vivo studies. So, what is new in my approach? What are the novelties present in my research? The nanoparticles used for drug delivery should be able to undergo biodegradation, and depends on the types of bonds present in the material backbone. The novel polymeric material I am going to use in this research has these easily degradable ester bonds in its backbone, and the degradation of these bonds increase once the particles get internalized into the cells through the endosomal system because of significant increase in the pH. And this polymer material has two blocks, hydrophilic and hydrophobic block. So, we can able to develop a stable nanoparticle system, and we can load our drugs into this hydrophobic block, and we can attach our ligands in this hydrophilic block. Further, the direct comparison of different ligands will reveal potential candidates for targeting hepatic stellate cells and collagen fibers, which can be adopted in future studies. The PET MRI system available in UQ is one of the few advanced preclinical facilities exist in the world. So, the imaging protocols used in this research can be easily transferred to a clinical setting. Further, the concept of using both collagen fibers and the hepatic stellate cells will increase the sensitivity of the staging and the diagnosis performed through non-invasive techniques. So, what? We believe the positive outcomes may change the way hepatic fibrosis will be diagnosed and treated in future. By detecting and effectively treating the disease in its early stage, we not only save a lot of money, we can also enhance the life quality of the people by saving them from the need of organ transplant. This research will also appeal for more multidisciplinary efforts to address clinical complications associated with fibroproliferative disease as a whole. Nearly 45% of deaths in industrialized nations are associated with some form of fibrosis in vital organs such as heart, lungs, kidney and liver. Collaborations will be initiated with pathologists having expertise in histology, and articles generated from this project will be published in high impact Tier 1 journals. Thank you.